Welcome to Lecture 3 of Biology 121, Human Biology and Evolution. I wanted to provide something for you to look at that's pleasing to the eye, while I remind you that you should be following along with the Lecture 3 cues found in D2L. This document contains questions highlighting the key information you should learn during this lecture. So, what you're looking at is Gustav Klimt's famous Tree of Life painting with its many wondrous, swirling branches. Because today we are going to continue our discussion about the biological tree of life. This idea that all organisms are connected to each other in a branching pattern of ancestry. We learned last class that this is called evolution. And today we're going to combine what we've learned about evolution so far with genetics, which is the study of inheritance. This image represents the molecule of inheritance that we call DNA. So in short, we are going to study the DNA or molecular record supporting the concept of evolution by natural selection. And this is meant to build on your prior knowledge that you were introduced to in the D2L lesson, Evidence for Evolution. So please take a moment for today's metacognition activity by finding a scrap paper and jotting down what you already know about these two questions. Number 1, list several lines of evidence that support the idea that evolution produces brand new species of organisms. 2, specifically explain the genetic evidence for evolution. Feel free to pause this video to give yourself time to jot down everything you already know. And then at the end of lecture, as always, we will revisit these two questions to see if I was able to explain any new details so that you can get your money's worth out of this lecture today. Briefly, I wanted to review what you already learned in the D2L lesson, these other pieces of evidence supporting the theory of evolution by natural selection. So one of the first lines of reasoning you considered was the fossil record. Remember that deeper fossils, ones dug up from deeper in the Earth's surface, are considered probably older, and carbon dating bears that out. By comparing fossils at different layers in the Earth's crust, we can go back in time as we dig down deeper into the Earth's surface. And we have several examples of transitioning forms. So here I'm showing the presumed ancestry of the modern day horse, going back stepwise all the way back to an animal that lived 55 million years ago called the Hyracotherium. This is one example of fossils demonstrating a gradual morphological change from an ancestral life form to creatures that look more and more similar to those that are alive today. And morphological, you'll remember, is a term used to describe the body form. So morphological traits are things you see and measure. It's like the shape and size of a body. This suggests a lineage of ancestors through the ages leading to its descendant alive today, which is the modern horse. The next line of evidence you've already reviewed in that D2L lesson comes from comparing the anatomy of things currently alive today. For example, you saw that whales and other things that live in the sea, mammals, they have a similar structure as other mammals' legs, but whales don't need legs because they live in the ocean. Common patterns of bones observed among species who now live in wildly different habitats suggest shared ancestry if they all inherited that bone structure from the same ancestor. Here is an example of that. In the middle, we have limb structures of turtles, dolphins, horses, humans, chickens, and bats. And they're color coded. All the different bones, the humerus, the radius, ulna, the carpal bones, the metacarpals, the phalange bones, they're color coded, and what we see is that they're all present in all of these different creatures. So the logic is that the ancestor that lived in ancient times, that was the many great great grandparent of humans and dolphins and all of these creatures, had a particular limb morphology, had this bone structure. So instead of reinventing the wheel over the generations, as animals started living in particular habitats, evolution just tweaked that ancestral design, that ancestral bone conformation. And it allows dolphins to have an appendage that allows it to swim through the water. It allows humans to be baseball players. It allows horses to run. Then I have a quote down in the left-hand corner that says, "Evolution acts like a tinkerer, not an engineer." What this means is that the bone conformation we inherited is what we're stuck with. And you can see that it was just adjusted for animals living in different environments instead of being designed from scratch each time. A dolphin may prefer to have a slightly different body plan than it does because that might be even more efficient at gliding through the water. Or a bat's wings could be designed by an engineer to require less energy to fly through the air. But both dolphins and bats are stuck with this evolutionary heritage, this bone conformation that was tweaked to allow them to live in their environments but isn't really perfect. So evolution acts like a tinkerer, adjusting previous versions instead of starting from scratch like an engineer would design. New adaptations are created by making small modifications to existing traits, not by starting from scratch. This is called path dependence, and in D2L I've copied the link that you see here. It is an interview with University of Michigan medical doctor Randolph Nesse, and he describes path dependence in human evolution. So you can watch that. It's an optional watch. The next piece of evidence supporting the theory of evolution by natural selection comes from comparing embryos of different types of animals. And it turns out that all organisms look alike, very similar, during early stages of life. And we subsequently go through similar stages of development. And it's only later that we start looking like animals that belong in vastly different habitats. This also suggests shared ancestry if we inherited our developmental stages in our very, very early embryological morphology from that ancestor. So here's some drawings of embryos. And you'll notice that on the left, it's very difficult to distinguish which embryo is going to grow up to become which adult animal. Indeed, human embryos look nearly identical to sheep, to cow, to pig, to chicken, to turtle, to salamander, and even to fish embryos. These are all vertebrates. It's not until later stages of development that our body plans become specialized and more apparent, matching what we're actually going to grow up and look like. So despite big differences in adult bodies, all of these earlier embryos look the same, and we think that this is because the challenges of the embryonic environment are very similar depending on if you're a human embryo or a salamander embryo or a fish embryo. And so that developmental plan in the earliest stages of life hasn't been molded by evolution in the same drastic way that our adult forms have been. All right, now for the piece of evidence that we're going to take a deep dive into today, the molecular record. And this is talking about the genetic evidence supporting the theory of evolution by natural selection. Genetic sequences of closely related species are more similar to one another than are the genetic sequences of distantly related species, which also suggests shared ancestry. Let me break this down for you. Oh, but before I do that, I wanted to preview our lab activity for today. You're going to look at actual DNA sequences from these 5 animals and also humans, only I haven't told you which sequence comes from which creature. So you're going to go down the letters of the DNA and proofread them and count up the number of differences between sequences, and you're going to group them together based on their similarities and differences and try to see if you can guess which creature that DNA sequence came from. Just like the fossil record documents transitioning forms, the molecular record documents transitioning genetic sequences. And there's 3 types of genetic information found in animal bodies. You've probably heard of DNA. That's the molecule of inheritance that parents pass on to their children. But you can also study the molecular record in these other molecules, RNA and protein. So scientists have studied all 3 to guess at the relationships among animals, but today we're going to focus on DNA. But all of the stuff we're talking about could similarly be done with either RNA or protein sequences. Just a little review about DNA. The molecule itself is shown here. It looks like a twisted ladder, and the rungs of the ladder are made up of something called nucleotides, and there's 4 flavors of nucleotide that match in a predictable way. Now, I've included a D2L lesson about genetics so that you can review these details, but I'm just going to talk about it only a little bit in the lecture. So there are these nucleotide sequences that make up the rungs of the ladder, and they come in 4 flavors, which I will call A, C, T, and G for short. DNA is passed from a parent to a child, and it's the sequence of those A, C's, T's, and G's, the order at which they make genetic words called genes, that gives each organism its unique mix of traits. DNA also accumulates mutations every generation. So every person born has some mutations that popped up randomly that they didn't inherit from their parent, that it is a random error in the production of DNA that goes into egg and sperm, and so it's a mutation that's unique to that individual when they're born. And we all have these. Some DNA sequences are identical between 2 people because they're related. So for instance, some DNA sequences are identical in siblings because the siblings have the same parents. If we extend this logic further out in the family, some DNA sequences are identical in you and your cousins because you both inherited that particular sequence from your shared grandparent. Similarly, if we extend this logic out even further, some DNA sequences are identical in different species of organisms, like humans and chimpanzees shown here, because those sequences were inherited by both organisms from their shared many great grandparent. And we call this great grandparent the last common ancestor of chimpanzees and humans. LCA, last common ancestor. Okay, so I talked a little bit about mutations, which is a change in the letters or the nucleotide sequence of DNA. In this example, a C mutated into a T, and that happens randomly. And the speed at which it happens randomly tends to be static, so we know how many mutations we expect per person, but certain environmental factors contribute to more mutations happening than is normal over time. And mutations occur randomly. And they occur more often if exposed to things like chemicals that we call mutagens. So this is a fictional example. Spider-Man was exposed to some mutagens in a horrible accident and then got all of these superpowers. But a real life example is when pregnant women smoke cigarettes. There are mutagens in cigarette smoke that cause her fetus to have more mutations. It is born with more mutations than it otherwise would have been as a result of her smoking. And mutations are interesting because they drive variation. And remember last week how I said Darwin was really interested in variation? Well, now we can study genetics, which Darwin didn't know about. Mutations are the source of new variation, and we call that new genetic diversity. And because they happen randomly, mutations may or may not affect survival and reproduction. Most mutations go unnoticed, and we call those neutral mutations. They pop up, we don't really notice, we go throughout our life, we pass them on to our children, and it doesn't affect us. Some mutations can be disadvantageous, meaning they inhibit survival and reproduction. They have bad effects. And some mutations can be advantageous, like lactase persistence mutations that you learned about in lab last unit. But most mutations are neutral. Over generations, mutations accumulate in the DNA sequence in that lineage. So imagine a traditional couple, they get together, they have some kids, their kids grow up, have some kids of their own. We have these very traditional family systems here, producing lots and lots of grandchildren and great-grandchildren. Imagine if, by random chance, a mutation occurred in this woman. That mutation doesn't go away when she has children. In fact, it gets passed on to all of her descendants. There they are. In this same lineage, maybe a second mutation happened in this person by chance, and she passed it down to all of her descendants. This is the interesting part. If you, as a scientist, came around and studied this great-grandchildren group of people, and you knew nothing else about their ancestors that came before them, and you collected blood just from these 1, 2, 3, 4, 5, 6, 7, 8 children, great-grandchildren, you could learn about their ancestry by comparing their DNA sequences. And how you would do that is by grouping the ones that are alike and suggesting that the ones that are most similar to each other are the ones that are most closely related. So if you were to do that, how many groups would you lump together? Let's see. You'd have these 4 together because they lack both the red and the blue mutation. Then you'd lump these 2 together because they have only the red mutation. And you'd lump these together because they have both mutations. And you'd say that the 4 in the big circle are closely related to one another, and the ones within each little circle, well, they're more closely related within the circle. And you would be right. And from that, you could accurately decide who is most closely related to whom. And this could be done, let's say all of these children were adopted, they could find their birth relatives. But if you extend that logic out, we can do that between different species as well. Here's what it looks like on the molecular level in the letters of DNA itself. So here's some original DNA sequence. The next generation was born and that C mutated into a G. Now the next time that this person has children, that G doesn't revert back to a C, it's a G forever. So they have children and eventually in the lineage, another mutation randomly occurs. Now a G turns into a T. That's inherited, it's passed down, and now we have a third mutation, this C in the middle. And so on. There's another mutation. So over time, we get a gradual change in the DNA sequence itself. Now this we call microevolution. It's a form of evolution because the genetic trait is changing over time. And at the end of these generations, what we get is a sequence that is similar to, but is not identical to the original ancestral sequence that we started with. Now imagine this happening in multiple lineages at once. So here I have a parent who had 2 children, only one of whom had a random mutation right there. The original C mutated into a G. This could be like me and my brother. And let's say my brother and I have a bunch of children apiece, and we pass on our genes to the next generation. Now every row represents a new generation, so kids and grandkids. The point is that these 2 closely related lineages are each accumulating separate sets of mutations. Over time, they become less and less alike, but they still have a lot of overlapping identical bits. And we see that, for instance, the ACT at the beginning of the sequence, it matches in both lineages and it matches the ancestor. So if we collected genetic evidence, like from a blood draw of animals alive today, and they have a bunch of DNA bits that are identical, we think that they're probably closely related species. Maybe they're cousin species that shared a recent common ancestor. And the more mutations that differ between any 2 animals, the logic is that we think they're more distantly related. So more differences, more distantly related. More similarities, more closely related. Eventually, with enough mutations, 2 lineages can become distinct species, and we call this speciation. An example of this is brown bears and polar bears. They're as closely related as bears can be. Only a handful of genetic mutations accounts for all of the behavioral and physical differences in these 2 species of bears. They're very, very closely related, and we know that because they have nearly identical genetic sequences. So here is a family tree of bears, and we see our brown bears and polar bears up at the top. They're as closely related as 2 bears can be. If you follow their blue branches on their family tree down, we see that it meets, and this point at which the branches meet is called a node. And the length of the branches downstream of the node represents the amount of time that these 2 bears have been separate lineages accumulating their own mutations. So here is a red dot and here's a blue dot, and these represent the mutations accumulating in the separate lineage of bears. Similarly though, in this bigger bear family tree, other mutations popped up a long time ago and have been inherited by many of the downstream ancestors. And that's how we know the mix of mutations. If we looked at all these bears' DNA, that's how we know where they belong on the tree tips of this bear family tree. You can use genetic analyses to place different species of organisms on a tree based on the level of similarity in their genetic sequences. And when you do that, it is called a cladogram. This cladogram is similar to the bear family tree in that time is flowing from bottom up. So that present day species alive today go here at the tippy tops of these branches. And if you follow the branches back, each connects at a node. So here's all species alive today. Here is a node that connects just these two species. That node represents the last common ancestor of those two species, and it existed somewhere back in time. If we go even further back in time, here's a node, and from that node, these three species eventually evolved. And the nodes keep getting deeper and deeper in time, connecting larger and larger groups of organisms. Eventually, we get all the way back here to the node that connects all of these organisms. And this moment in time is when we think the last common ancestor of all 6 of these organisms must have lived. And if we know the rate of mutation, the average rate of mutation per generation, and we know the average generation time, like for humans, we assume it's about age 20 when people have their first kids, so we estimate generation time to be 20 years. Well, it's different if you're talking about mice versus dogs versus elephants versus humans. But depending on the species, we know the rate of mutation per generation, and so we can look at the number of mutations and multiply that by the generation time to estimate how far back in time we think a last common ancestor lived between any two groups of organisms. Isn't that interesting? So for your lab activity this week, you're going to look at several different animal species that are alive today, including humans. You're going to try to identify them based on their genetic sequences and your prior knowledge of these species. So let me tell you what they are, starting with the guy in the upper left-hand corner with the fabulous red hair. That's an orangutan. To the right, that is a rhesus macaque, which is a type of monkey. Then there's a sheep. Below the sheep is obviously a human, and that's Tucson's mayor, Jonathan Rothschild. To his left is a kangaroo rat, which is a rodent. And then in the bottom left-hand corner, that is a mouse lemur. And you may or may not know anything about these species, so when you're doing your lab, just make your best guesses. And then I'll have to tell you what the correct answer is. Maybe in the next video I'll reveal which sequence belongs to which animal. Oh shoot, I did that already. Spoiler alert. I thought I deleted this slide, but I didn't. So those are the animals and how they're related on the cladogram, and the sequence number is above their heads. All right, so the key points from today's lecture so far are that related species share some identical sequences of DNA because they inherited these sequences of DNA from a common ancestor. Species that are more closely related to one another had a common ancestor more recently in time. Genetic sequencing gauges how closely related different species are to one another. And genetic differences may be observed in DNA, RNA, or proteins. If you're interested in this topic, you could read a popular science book called The Making of the Fittest by Sean Carroll. Or you might watch episode 102 of The Cosmos with Neil deGrasse Tyson. It's available on YouTube. I watch it every now and again. And he has a really cute example about how dogs evolved from wolves. That brings us to today's case study, titled Murder by HIV. And this one is a really interesting real-life court case that happened in Louisiana after some tragically bad behavior by a medical doctor against his former lover. And I wanted to share some information about this to set you up for the case study. Here is a YouTube link of some sensationalized news coverage of this doctor's parole hearing. And here he is. His name was Richard Smith. He was a medical doctor on trial for attempted murder. The motive was a lover's quarrel. He was charged for the attempted murder of his ex-girlfriend. And the weapon was an HIV contaminated B12 shot. So the history of this couple is that she had B12 deficiency, and throughout the relationship it was normal for him to give her B12 shots. However, one day he came in, she was actually asleep, and she was abruptly woken up when he stuck her with a needle and said it was a B12 shot. And that was abnormal behavior, she reported. So we have the motive, we have the weapon, and the opportunity is that this doctor worked with many HIV positive patients, as most doctors do. And like I said, he was in the habit of giving his girlfriend B12 shots. But what was suspicious about this particular potentially contaminated B12 shot that supposedly was the attempted murder weapon was that he had a patient, an HIV positive patient, whose blood had been drawn the same day that he injected his girlfriend in this weird way when she was asleep. And that patient's blood work mysteriously disappeared. And I don't know if you've ever had blood work done or if you've worked in a lab, but this is monitored meticulously. There are labels on everything, so it is really abnormal for blood work to go missing. And for it to happen on the same day, that was suspicious. So here's some evidence. The doctor drew blood from one of his HIV-infected patients, and that sample got lost. There was improper documentation, and as I said, that is a big no-no. Thankfully, it's rare. Later that same day, he gave his ex this B12 injection that she didn't consent to. Like I said, she was asleep, and she woke up getting a needle jabbed in her body by her boyfriend. So very suspicious. She later found out that she was HIV positive. Now, his girlfriend was a nurse that actually he had employed, and they had had a many-year-long tumultuous relationship. They were both married to other people at the time, and you can read about all the details in the case study. She had been tested as a nurse for HIV many times, and always it came back negative until one time it came back positive. So she'd formally tested negative. The way the law was set up in Louisiana at that time is that when someone newly became HIV positive, they were set up with a representative of the health department who helped them contact and required them to disclose and contact all of their former lovers. So she had to go through that with a representative of the Louisiana health department, but thankfully all of her former lovers tested negative. So that ruled out that she contracted the disease from them. Here's where it gets interesting to biologists. The prosecuting attorney called a molecular biologist expert witness. HIV is an RNA virus. So RNA sequencing was done on strains of HIV found in the victim's body. And this sequencing revealed that the victim's HIV strain was not identical to, but was a close match to that of the exact patient whose blood had gone missing from the doctor's office that day. Now we know from virology that HIV mutates rapidly, and the trial was years after the alleged event. However, the molecular biologist expert witness said that strains of HIV found in the victim and that patient whose blood had gone missing, they shared a common ancestor, in fact, around the time of the alleged event, that fateful day where the patient's blood went missing and the victim was shockingly injected by this doctor. These strains of HIV lumped together in a cladogram, while other local strains from different people in her community, in the victim's community, they were on different branches of the cladogram. Here's what the actual cladogram presented at that trial looked like. Let me explain what you're seeing here, because it looks like a mess at first, but we can break it down. All of these sequences with names that begin with P were found in the blood work of patient 0, the patient whose blood had gone missing. And these were all different sequences of DNA found in that person's body. And so you can see that any HIV patient, they don't have just one strain of HIV, because HIV mutates so rapidly that as soon as a person is infected, it mutates instantaneously, and there are 2, then 4, and then multiple different strains of HIV going on in one body. And like we said, the trial was years later, so it makes sense that there's all these different strains of HIV in patient 0. In the middle here, all of the sequence names beginning in V were found in the victim's blood at the time of the trial. And as I said, because HIV mutates so rapidly, it is normal to find many different strains within a single person. Now, all of these other sequence names down here, they begin with the letters LA for Louisiana. These were collected from HIV positive people living in Lafayette, which was where the victim and the patient and the doctor all lived. So these are community strains. And what they're doing here is trying to rule out that the victim might have gotten infected by one of these other people. And this cladogram is tilted on its side, where the other one we had the bears and the organisms, that the branch tips at the top. Here it's tilted so that we have all these strains over on the right side, and that's fine. That's just an artistic preference. But you can still trace the branches back to different nodes. And if you trace the P branches backwards, and that's to the left, and then you have another finger on the V branches, you can see where their node intersects. And that would be the moment of infection. So if we continue that logic, we could see who patient 0 was infected from, and actually it looks like it must have either been from LA08 or from LA32. And so in any community, you can trace the spread of HIV in this way. Interesting. So that's how those strains lumped together in a cladogram. And the verdict from the jury was that this guy was guilty. They gave him 50 years in prison. And he suggested that this is a new type of evidence. It shouldn't be part of legal precedent. However, when he appealed all the way up to the U.S. Supreme Court, his appeal was denied, and that established the molecular record as legal precedent for criminal cases here in the US. So that's a score for the biology nerds. And for the logic of cladograms and genetic sequencing. All right, I hope you enjoyed that sensational and yet true story, Murdered by HIV. Please read the case study in your free time. So we're done with the molecular record portion of today's lecture. So take a minute to review what you wrote down at the beginning of the video and see if you can add anything to your list. And I'll pause for a few seconds. Feel free to pause the video. Okay, lab this week. Lab for Unit 3 is rather time consuming because it introduces the concept of genetics, and genetics is a big concept. So the first activity, you're going to view how parents pass on genes to their offspring. The second activity, you're going to see how DNA inspires your phenotype through the production of proteins. And then the third activity, you're going to evaluate how natural selection has acted on a human mutation to maintain the genetic disease called sickle cell anemia, which is very common worldwide and particularly here in the US. And this last activity, I find it so interesting, and there's a video attached to it, so there's a link to that in the lab instructions in D2L as well. And I hope that you find it interesting too. Here's the link to the video, but like I said, it's also in the lab instructions in D2L.